Hello everyone, good afternoon. Welcome to our 25th edition of the INFOGEST Viva Webinar. My name is Sara Perez Vila and I would like to introduce you my colleagues Daniela Freitas and Nikki, who will be conducting the webinar with me. This event is organized by the team of postdocs from Teagasc Research Centre in Ireland. These webinars aim to share the latest findings in digestion with presentation from the most recent graduate PhD, postdocs and younger researchers. These webinars are broadcasted live in YouTube and will be accessible in the InfoGest YouTube channel. The presentation is a pre-record video, which will be followed by a question and answer session. So I would like to invite you to type our questions and answers in the bottom at the bottom of the screen. Our speaker today is Dan Liu with her project about untangling the interplay between food microstructure, mechanical properties, microstructural breakdown, and in vitro gastric protein digestion. Dan Liu is a young researcher working on food protein digestion. She just received her PhD in February 2025 at Wageningen University. During her PhD, she has explored interplay between food microstructure, mechanical properties, microstructural breakdown, and gastric protein digestion. So, Dan, the floor is yours.
Hello everyone, I'm Dan Liu. I got my PhD at Wageningen University earlier this year in February, and now I'm continuing here as a postdoc. I'm very happy to have the chance here to share the work I did during my PhD with you. We all know that as a key macronutrient in our daily diet, proteins are crucial for the growth, maintenance, and the metabolism of the body. For decades, humans have relied on animal-based foods to obtain sufficient protein. But as you may also know, the production of animal-based protein foods requires a lot of resources and greatly contributes to greenhouse gas emissions. Therefore, meat analogues have been developed with other plant-based protein sources using various technologies. On the one hand, the nutritional value of meat analogues is challenged by the low quality and digestibility of plant-based proteins. On the other hand, the introduction of new technologies offers great opportunities to optimize the nutritional properties of foods by engineering food texture and structure. For food structures, they can strongly influence gastric digestion. These structures include oral macrostructural breakdown, mechanical properties like the hardness or the stiffness of the food, and the microstructure. But as you can imagine that these food structures are often closely connected to each other, making the impact of food structure on gastric digestion very complicated. Therefore, with this project, we aim to untangle the interplay between food properties and in vitro gastric protein digestion. To achieve this, we planned 4 studies, and for the first study, we investigated the independent impact of macrostructure, mechanical properties, and macrostructure using model gels. So we made these whey protein gels, and they all had the same protein content, the similar Young's modulus, so the same hardness, and the same surface area, which comes from the same macrostructure. The only difference is we add a small different amount of kappa carrageenan to the gel solution, which brings the gel distinct macrostructure. As you can see on the screen, the red part refers to the protein-rich phase, and the dark part refers to the non-protein-rich phase. So we had homogeneous, protein-continuous, cross-stranded, and bicontinuous macrostructure. Now we put these whey protein gels with different macrostructure into the in vitro gastric protein digestion model. We used a static INFOGEST protocol, and then we measure the free amino group concentration during the digestion, and based on this, we calculated the digestion rate or the free amino group release rate. So for the result, we found that the homogeneous gels showed the highest digestion rate, followed by protein-continuous, cross-stranded, and bicontinuous gels. This means that the gel macrostructure itself can affect in vitro gastric protein of whey protein gels. Then we studied the independent effect of mechanical properties. We started with homogeneous gels. So we had these two gels with the same homogeneous macrostructure, but different Young's modulus. So one gel is harder and the other one is softer. As expected, the softer gels was more digested than the hard gels. This means for the homogeneous gels, increasing the Young's modulus or the stiffness could hinder the in vitro gastric protein digestion. Then we also tried with protein-continuous gels. So we had these gels with a series of Young's modulus, and they all showed protein-continuous macrostructure. However, there was no significant differences in free amino group concentration during static digestion, which suggests that for the gels with protein-continuous structure, increasing the Young's modulus or the stiffness did not affect the in vitro gastric protein digestion. This result shows that the impact of mechanical properties on protein digestion depends strongly on macrostructure. We also studied the independent effect of macrostructural breakdown. We simply cut the gel cylinders into many small gel cubes to roughly mimic the oral breakdown. And the only difference between the gels is the total surface area caused by cutting. As expected, for all the gels, the cubes were more digested than the cylinders due to their larger total surface area after macrostructural breakdown. But if you look closely to the results, you will find that for the gels with different macrostructure, even though they had the similar increase in surface area, the bicontinuous gels showed a larger degree of increase in digestion rate compared to the other three gels. This suggests that the effect size of macrostructural breakdown on protein digestion also depends on macrostructure. Then we realized the important role of macrostructure in protein digestion. But then we wonder, okay, what if these gels are increasingly or intensely crushed, just like when we chew them? So for the second study, we explored the impact of macrostructure after oral macrostructure breakdown. So we still have these whey protein gels with different macrostructure. We asked a group of people to chew them following a fixed chewing process. Then we collected the bolus, and we also analyzed the bolus particle properties, such as bolus particle number, particle mean size, and total bolus surface area. Then these bolus were subjected to the in vitro gastric protein digestion. After mastication, both the free amino group concentration and the digestion rate were increased compared to that of unmasticated intact gel cylinders. This is because the great increase in total surface area which provides more spots for pepsins to do protein hydrolysis. Regarding the degree of increase in digestion rate, we assume that after chewing, the larger the degree of macrostructural breakdown, the higher the digestion rate. For example, the gels with more and smaller bolus particles would be more easily digested compared to the gels with less and larger particles, even though these gels differs in macrostructure. However, the results showed that there was no correlation between bolus particle properties and digestion rate. For example, if you look at the first figure from the left, you can see that the cross-stranded gels have more bolus particles than bicontinuous gels, showing a larger degree of structural breakdown during mastication. But the cross-stranded gels still show a lower digestion rate than the bicontinuous gels. And for bolus particle mean size, we did not find that the smaller the particle size, the higher the digestion rate. Or for the total bolus surface area, it was not like the larger the total surface area, the higher the digestion rate. This means there is no correlation between macrostructural breakdown degree and the digestion rate. And remember, the only difference between these gels was macrostructure. So we concluded that the effect of macrostructure on protein digestion is sustained even after oral structural breakdown of mastication. So far, I showed you that when it comes to protein digestion, macrostructure matters and oral macrostructural breakdown also matters, but all these are based on the studies on model gels. And I believe no one will have this for dinner, and you probably prefer a plant-based burger. So when you are eating a plant-based burger, you are usually consuming textured vegetable proteins or TVPs. These TVPs typically have a porous structure to mimic the texture of meat. And by manipulating the process conditions, these TVPs can show different structural porous structures, and these structures can be different in many properties such as pore size, porosity, and wall density. In this study, we used 8 TVPs with different structural properties, and for each TVP, we had 3 treatments. So we ground the TVP into powders to remove the porous structure and as a control group. And we had the original TVPs, and we also made plant-based patties from these TVPs. Then they were subjected to the static digestion model to perform in vitro gastric digestion. We quantified 8 structural properties of TVPs, then we correlated them with the free amino group release during digestion. We got many results, but for the sake of time, I will just show part of them. So we found positive correlation between mean pore size and free amino group release for TVPs and for TVP patties. This means the large pores in TVP structure facilitate the protein digestion of TVPs, and this impact is sustained for TVP patties. We also found a negative correlation between wall density and free amino group release, but this correlation was only there for polars. This means that the wall density affects protein digestion of TVPs only when the porous structure were removed. So these results suggest that pore size contributed more to the proteolysis of TVPs and patties than wall density, and for enhancing protein digestion of TVP-based meat analogs, we suggest increase the pore size of TVPs with a porous structure and decrease the wall density of TVPs with a compact structure. But I believe no one's stomach looks like this. It looks more like this, with gastric peristalsis and gastric empty. Therefore, we were interested in the dynamic digestion, which can mimic the gastric motility. This time, we focused on the interplay between mechanical and bolus properties. So we have 2 commercial plant-based patties, and the Beyond Meat patties were less stiffer than these patties. As a result, after a fixed trim process, the Beyond Meat patties showed a larger bolus particle size than these patties. Now we put the boli of these 2 patties into a static model and a dynamic model which can mimic gastric fluid secretion, gastric peristalsis, and gastric emptying. For both models, we measured the free amino group concentration during digestion, and for the dynamic digestion, we also measured the gastric content to evaluate the in vitro gastric emptying. For the results, for the static digestion, we found that Beyond Meat patties were more digested than these patties, and we believe that the lower Young's modulus of Beyond Meat patties contributed to its lower digestion. This suggests that the impact of Young's modulus on protein hydrolysis was more pronounced during static digestion. However, for the dynamic digestion, we observed the opposite result. So during dynamic digestion, the Beyond Meat patties was less digested than these patties. And this is because the larger bolus particle size of Beyond Meat patties slowed down the in vitro gastric empty, which eventually hindered the protein hydrolysis. So from this study, we found that the bolus particle size primarily impacts the dynamic in vitro digestion of plant-based patties, and the Young's modulus mainly impacts the static in vitro digestion of plant-based patties. And this study also highlights the importance of methodology in vitro studies. Overall, I hope with these studies, we showed that designing macro and macrostructure of food is an effective approach to modify protein digestibility. And for food scientists, we should select our in vitro digestion methods thoughtfully based on research questions. Thank you for your attention, and if you want to know more about my research, you can scan the QR code to read my PhD thesis online, or you can contact me via LinkedIn or email. Thank you very much.
Okay, thanks. Thanks very much, Dan. That was a really interesting presentation, and you did a lot of work as well. I was wondering if you looked at this, or whether you think it would be interesting to look at how the particle size evolved, the size of the particles of the chewed boli evolved during digestion. For example, if you compare the static with semi-dynamic digestions, or sorry, the dynamic digestions.
Yeah. Hi, Daniela. So I think in my study, I think it's clear that for during the static digestion, it seems like the texture or the hardness of the samples, or in our case, it's a plant-based protein burger, plays a more important role than the bolus particle size. And during the dynamic digestion, I think we showed that it's a bolus particle size showed a more pronounced impact on the protein digestion. And I think it's because in our study for the dynamic digestion, we used a dynamic system which emphasizes the gastric motility. So we basically use a caloric valve to control the gastric. We don't control it, but it's like the smaller the particle size, the faster or the earlier it gets emptied from the stomach. So I think that's the main reason why during the dynamic digestion, it's a bolus particle size plays a more important role.
And do you know if by the end of gastric digestion in the NERD, in your dynamic model, the particle size was still significantly different? The particle size distribution was still significantly different between the two patties that you tested?
To be honest, I don't know because we didn't test it. Yeah, and yes, I just don't know.
Yeah,
Yeah, I don't know.
Yeah, I was just wondering whether this effect that starts during the oral phase and that has an impact on gastric digestion could also impact what happens after gastric digestion.
Yeah, I think there might be an influence because if the smaller particles get emptied earlier from the stomach, then it also enter into the intestinal phase earlier. Now it can be it can be further hydrolyzed. Then in our body there will be a metabolic response from this released nutrients. Then our stomach will also receive this feedback. It will behave differently.
Exactly. Yeah.
Yes.
And in the NERD, what else what else is different compared to the static? Because you have the progressive emptying, but also there are also other parameters. The flow of the gastric fluid, for example, is continuous. Is the pH also different, or the enzyme concentrations?
I would say everything will be different because compared to the static protocol, which is basically a beaker and then nothing goes in over time and nothing goes out over time. And during the dynamic digestion, first the gastric fluid secretion is different. We have changed secretion rate to mimic the to mimic what happens in the real body. And because of the empty, the gastric content is always changing. And because of this, you have I have no idea what's the enzyme concentration.
Oh, okay, but it's varying, a lot during.
Yeah, it varies a lot. Yes, everything, including the pH and enzyme. So it's really a dynamic system.
Yeah, it is very complex system. It's really interesting to see the comparisons between this one and the static, the digestions as well. Okay, so I think that we do not have any more questions. I don't know if no. Okay, we don't have any more questions in the chat. Okay. So thank you very much, Dan, for presenting your work and for doing such a good job with your presentation as well, and a very clear and nice slides. Thank you, and the best of luck with your next projects now.
Thank you.
Thank you. So I will maybe just share a slide to announce the next speaker. So now we have a break over the summer, and we will be back in September. Let me just see. Okay, in September, on the 3rd of September, with Nicolas Horlacher from the University of Otago in New Zealand. In the meantime, we wish everyone a good summer break. And we are also opening we have an open call for expressions of interest. If you would like to present your work in the next season, let's say, of the INFOGEST Vivas for 2025 and 2026, please email your name, affiliation, and suggested title of your talk to infogest.viva@gmail.com. So thanks very much for joining our webinar today, and we will see you again in September. Thank you. Thanks, Dan.